This is the series which celebrates alumni who have gone on to make remarkable contribution in science, research, and industry. And we are honored to have you with us. Thank you so much for joining. It's a pleasure to have you.
Well, thank you, Yash. Thank you for inviting me for this podcast. Good afternoon to you, first of all. And it's really a privilege to be connected with you and the other BCP students after a hiatus of so many years. So, thank you for the opportunity.
To start off, ma'am, could you tell us a bit about your role in AGOs and what does a typical research day looks like for you?
Okay, so we can start there. So, I've been in this role for about 5 years. I joined Geos Pharmaceuticals about 5 years ago. It's a small biotech, about 500 employees, and we are focusing on rare hematological disorders primarily. So when I joined the company, my role was to head the Department of Drug Metabolism, Pharmacokinetics, and Clinical Pharmacology. Over the years, I have also gotten responsibility for toxicology and the overall research side of the organization, which involves chemistry, biology, pharmacology, and other related functions that are required to discover a new molecule. So that's the current role that I serve. There is no such thing as a typical day, as you can imagine, but more or less it's the oversight of all the functions. If anybody needs any problem solving, talk to them, keeping in touch with mostly all my direct reports, but also all my team members to see how they are progressing in their job functions, their assigned responsibilities. And then because I also have a late stage role in terms of registrational filing, there is a lot of document review that I have to do. So many days I'm reviewing documents that are related to DMPK and clinical pharmacology.
That is fascinating. Ma'am, many students, when hear the term DMPK, they don't always grasp what it truly means. So, can you explain us in a simpler way about DMPK and PKPD in context of drug discovery?
Absolutely. DMPK stands for drug metabolism and pharmacokinetics. PKPD stands for pharmacokinetics and pharmacodynamics. Both these disciplines, so to say, play a vital role in throughout the discovery process. So starting from early stage when the molecules are first being synthesized by medicinal chemistry all the way to registrational studies. So there is a significant role to be played. What pharmacokinetics means is you probably have taken, I don't know, a few courses in pharmacokinetics offered by BCP. That is what the drug does to the body. How does the drug handle the body? And so you are understanding the absorption processes, the distribution processes, the metabolism processes, and the elimination processes. That is, in a sense, DMPK. Although that it's only focused on the M, it actually is much broader than that, in terms of how the body is handling the drug. The PD side of it is actually what is the drug doing to the body. So it has, you're making this new molecule because it has certain affinity, potency towards a particular target that you want to modulate in the body. So when the drug achieves certain concentrations in the body, what does it do to the body? How does it achieve the desirable efficacy that you want to achieve? Understanding that is the PD portion of it. And then when you put the two together, that's what you get, PKPD. Now, there's much more beyond that, in terms of because most of the drugs that we deal with are metabolized. There is also a high potential for drug-drug interactions with other drugs that are co-administered. So DMPK also involves study of that. The concomitant administration and what that might do to the PK of your drug or the PK of the concomitantly administered drug. For the last several years, there has also been a significant focus on transporters. These are I would say enzymes, for the lack of a better term. They're not quite enzymes. They're proteins that are actively transporting the drug from one side of the membrane to the other. And this becomes important if the drug is either a substrate or an inhibitor of these transporters, because what that can happen is these transporters can govern how much the drug can concentrate in the tissue. And if that tissue is where your site of action is, obviously that's going to affect the efficacy. But if that's not the site of action and the drug is concentrating in there, you can also lead to significant adverse events. So DMPK also deals with such phenomena, drug-drug interaction at the broader term. And so as you can just see based on the brief description I gave you, you can see how that can play a significant role throughout the process of drug discovery and development.
Definitely, ma'am. I think the students will now finally be able to connect with the concept, and it was much clearer and approachable explanation. Uh, when it comes to pharmacokinetics, all of a sudden we just know Krishna Iyer sir because they have taught us pharmacokinetics. So during your graduations, who were your mentor for this subject?
So I believe there was Professor Shargil who used to offer one class in pharmacokinetics. I remember that was like an introductory class that he used to offer. Uh, it was rather basic. That's the only class I remember. Most of the other classes were, what you have described to me earlier in terms of what the current curriculum looks like. We had pretty much the same curriculum, but we only had a 3-year program.
When did you realize that you have a more, interest towards pharmacokinetics?
So when I came to this country, I came in to do a master's in pharmaceutical chemistry, medicinal chemistry. And then after doing my master's, I was just exploring to see what field of interest seems appealing. And it seemed like understanding PK, drug metabolism, may be a place that I can enjoy. Again, not having a lot of background, I pretty much just jumped into it. I think it was just by, you can say inclination, you can say by chance. I applied to universities that were very well known for the discipline of pharmacokinetics. And that's how I chose University of Washington in Seattle. And I got the best training possible in DMPK from that school. So, and then I fell in love with it once I started studying, much more in depth, all the courses associated with DMPK.
That sounds amazing. You were also a part of journey for pyrukine's approval, which is a major milestone for AGOs. So, what made that project so special from a scientific standpoint?
Yeah. So let me tell you, I arrived at Agios in late 2020. Agios has been developing pyrukine for several years prior to that. So when I came, they were in the midst of putting together all the package for registrational filing. So as I came in, my job was to really take an overview of the entire package, see if there are any gaps, and really prepare the team for answering questions. I was a newbie on the block, really, so I relied a lot on the scientists that had been working on pyrukine. But what was really interesting to me was that this is the first time I had direct responsibility for clinical pharmacology. During my previous 28 years of my career at BMS, I was never directly responsible for late stage clinical pharmacology studies. So here it offered me the opportunity to get involved, not get involved, the studies were completed, but really to position ourselves from any regulatory questions. And as I said just now, this molecule, pyrukine, is a substrate, as well as an inducer of an enzyme called cytochrome P453A4, and that can lead to significant drug-drug interactions. So I think the most interesting part was how the studies were done to understand the risk, buy down the risk, and suggest to the physicians and patients how you can take this drug in presence of other CYP3A modulators, or how you can take this drug with other CYP3A substrate.
That must have been an incredible experience. When it comes to DMPK, PKPD, before a drug reaches human, there are DMPK studies that play a huge role. So, what do you think these studies help determine things like dose, formulation, and safety?
DMPK plays a significant role in that phase. Um, Very early on, when the molecules are first made, chemists are using, they're working with the biologist and they're using some models to understand what the target structure looks like and what kind of molecules you want to synthesize. And earlier, the very early studies just focus on the binding affinity of the molecule to the target. And then they progress towards cellular assays. Most of the targets are intracellular. So they will then assess the affinity of the molecule to the target in a cell. Okay. Now, once those studies are done, once you understand the molecules have sufficient affinity to the target, now you want to understand what the DMPK properties look like. So a lot of the times there are first in vitro studies that are done to assess solubility of these molecules, to assess cellular permeability of these molecules. Then we get into metabolic stability, because if the drug is extensively, rapidly metabolized, it's not going to have a very good half-life. We also assess the drug-drug interaction potential against various enzymes and transporters in vitro. Then comes, and of course, these are tiered. We don't do everything at once. Then comes the time when we actually put the molecule in vivo in an animal model and see what does its pharmacokinetic profile look like. So a lot of the times the first studies are done in a rat because it's easier to draw the blood samples, as you can imagine, and generate the full pharmacokinetic profile. But many times the efficacy studies are done in a mouse model. So then DMPK scientists will assess the PK profile in a mouse, because then that gives them an idea what kind of plasma concentrations you reach after a certain dose. Sometimes early on, these molecules just do not have the right properties to be orally absorbed. But because these are novel targets, you really don't understand what this target is going to need for its modulation. So you might choose another route, such as an intramuscular, intraperitoneal route, whereby the barrier to get in the systemic circulation is less relative to the GI tract. And so the aim of these DMPK studies is to understand the PK profile, what concentrations you achieve after a given dose, how to modulate the oral bioavailability. You could then try different formulations that you are very familiar with to try and see if you can improve the oral absorption. And then what kind of dose, what kind of dosing regimen you might need in order to achieve that efficacy in an animal model. To give an example, if you're talking about a cancer, an anti-cancer molecule, you may have an animal model in a nude mouse most of the time for cancer drugs, where you're implanting a tumor and you're looking at how the growth of the tumor is suppressed after chronic dosing of the drug. So you want to administer the drug more or less daily, since it's a chronic administration, and look at its PK profile and look at what it does to the tumor efficacy, and then change the dosing regimen accordingly in order to see what you want to see. What is the desirable efficacy look like? You may say that some of these drugs don't work at all. You have no efficacy. So you go through at least tens of molecules in vivo, but hundreds of molecules in vitro before you get to this in vivo stage. And as you can see, DMPK scientists are working really in close collaboration with these other disciplines, such as medicinal chemistry and biology and pharmacology, to get to the proof of concept in an animal model. Okay, that's the very first stage in discovery. And then there are studies that you do beyond that. Once you hone in on what molecules look interesting to you, which of these molecules you want to advance further to development.
That's amazing, ma'am. And it really shows how DMPK shapes every early decision in drug development. From your experience, what are some common misconceptions that students have about, DMPK? So how can they begin exploring this field in the right way, as you have decades of experience in this?
As I was talking to you earlier, there is a DMPK online course that is offered by SSX India. It is offered every year. And I believe there are about 25 different classes on different subjects that are part of that curriculum. And every lecture is given by a different person, different scientist. Many of them are from India, some of them from abroad, US, etc. Some from UK, and they cover all these disciplines that I was just alluding to. Drug metabolism, drug absorption, PKPD, animal models, etc. So that would be a good place to start to really get to understand what this discipline is about, because I don't, and there may be some universities that have courses in DMPK as well, maybe like a graduate level classes. There are a lot of misconceptions, but they arise mainly because there is no training in DMPK. So I would say that the course may be a good place to start. And then after that, you just like you, right? A lot of BCP alumni probably are in this field. Reach out to those, your alumni, and if you want to get any further clarification, talk to them if you're interested. As I said, misconception is because you don't have the fundamental training. That's really what it is. But it's a very interesting discipline, and uh, a lot you can do to contribute to the field of drug discovery and development.
That's a wonderful advice, ma'am. Thank you so much for it. You have worked extensively on transitional AD, ME, and PKPD modeling. Could you share how these models help bridge preclinical data to predict human response?
Yeah. So I was just telling you earlier, right? You have to optimize the dose and the dosing regimen in order to get a certain pharmacokinetic profile. And you're relating that to the efficacy in that animal model. Now, give you an example of tumor growth suppression. But a lot of these targets meaning what the molecule is attacking, in terms of the enzyme, etc. There's also, as the molecule interacts with the target, let's say there is an inhibition of certain kinase. Okay, many of these cancers are driven by overexpression of certain kinases. So when that kinase is inhibited, something happens as a result of that, like an immediate result of that, some protein will go down that the kinase makes. Okay, now that's a biomarker. You can measure that biomarker uh, in the tumor, for example, but many times you can also measure it in plasma, in the blood compartment. So that's a biomarker. Now, the DMPK discipline also measures this PDE parameter, this pharmacodynamic parameter, and try and relate the plasma concentrations to the pharmacodynamic effect, and then relate that to overall efficacy in terms of tumor growth suppression. This mathematical model that we built helps us now to project what might happen in human. Now, of course, you are still translating from mouse to human. So you have to understand, is the target the same in the mouse versus human? Is the affinity of the molecule to the mouse target same as the affinity of the molecule to the human? How are you going to translate the mouse pharmacokinetic profile to human? And how you're going to translate the pharmacodynamic profile from mouse to human. So all these things you assess, a lot of these things you assess in vitro. You build the pharmacokinetic models, and that's what helps you in selecting the human efficacious dose once you fully understand how these molecules of interest work in a mouse model. Of course, for pharmacokinetics, you will also study the profile in other species, such as the rat, and then higher species such as the dog. You're not just going to rely on the mouse, because not one species is a perfect replica of human. So you have to try and understand the behavior in multiple species and try to make correlations to what human may look like.
That sounds amazing. It really shows how science and data really complement each other, in the decision making. Talking about research, you've contributed to several publications and discussions. So, over the years, which research area has fascinated you the most?
I think I'm more interested in PKPD and trying to relate that to predicting human dose. So some of my papers that involve understanding the efficacy, as I was saying, in a mouse model and translating that to human, that probably is my favorite aspect of this science. But what I like about it is because you're building all these properties in the molecule before ever going to human. You don't know what this molecule is going to do in human. So trying to buy down the risk as much as possible so you do not have significant surprises in human. That to me is the most fascinating part. Designing, building the right properties in a molecule so you advance the right molecule. Because once you are in human and you start to do clinical studies, you have invested in that molecule so much. Beyond that, you are characterizing it, how it behaves in human, and then of course in the subsequent phase 1, phase 2, phase 3 studies. But you're not going to change the molecule. The opportunity to change the molecule and select the right molecule is in the discovery space. So to me, that's the most fascinating part.
Great, ma'am. In industry, balancing a day-to-day R&D with publishing can be challenging. So how do you balance this?
I have had number of publications. A lot of them are because I was in a position where I had large groups. So, anytime my group published, my name was there because I was somehow involved in the research. What you're doing in industry is good work, good research. You are using new technology, and you are making that knowledge available to the general public, right? The scientists, the scientific community. I would say that yes, you have to find the balance. I think it is important to publish because it teaches you, first of all, to be abreast of the literature and then see what your work can add to what's already out there. So that training to me is tremendous. Yes, it takes a lot of effort. And if you're working in an industrial environment, there are a lot of restrictions in terms of what you can and cannot publish. Because many of these drugs that the work that you're doing is patent protected, so you cannot publish until your IP is secured. A lot of times the companies just don't want the competitive knowledge to be out there, so that puts a lot of restrictions on what you can publish. But I believe that there is always an opportunity to publish, especially because as the drug clears certain stages, as the drug, let's say, reaches a phase 3, and especially small companies, they really want to showcase what they do, what good science they do. So they actually encourage many of these studies to be published. And I would say take advantage of that. Now you're publishing your own work that you did on the job. So that's not so difficult. What is difficult is when you're trying to publish something outside, what you do daily, and then you have to do a number of, reviews and then put together whatever, publication. But this is what you have done, in your daily job. That becomes a lot easier to publish. You can publish what you have been doing, as part of your job. And I would say just find that opportunity when is the right time strategically for the company to publish the work and take advantage of it.
I'm sure our students would, read more and more research papers and, newsletters and everything. Have led diverse scientific teams across locations. So in your experience, what makes a research team truly effective?
Yeah. I mean, especially with geographical distance, right? It makes it harder. I think really for the teams to work efficiently is the collaboration, right? I mean, DMPK alone is useless. It's not, what is a DMPK scientist going to do by themselves? They need in a discovery preclinical space, they need to work with medicinal chemists, they need to work with biologists, they need to work with toxicologists and so on. In the clinical space, they need to work with bioanalytical scientists or clinical scientists, or let's say even the commercial folks and regulatory folks. So collaboration is key.
Absolutely, ma'am. And what do you think are the qualities that our students should start building in order to get into this field?
Curriculum that BCP offers, that they have offered throughout these years, is very diverse. So as you know that you take courses in so many different disciplines, that's a great place to start, that you are exposed to so many subjects, right? try to understand these subjects, try to get deeper into these subjects depending upon your liking, where do you want to concentrate more, because that's going to shape, what you want to do after your BPharm. Be very curious, ask a lot of questions, read a lot of literature, talk to different people. Talk to your professors, talk to your alumni, talk to your seniors. I think I just build a love for science is what I would say. It doesn't matter what your discipline is. Build that love, and once once the science starts to fascinate you, I think you will do the right thing to sort of fulfill your appetite that you've created, and you will master whatever field that appeals to you, and then you will make a career out of it.
Definitely. Looking back, how did your time at BCP prepare you for this research-driven environment, as you have done a PhD in University of Washington? So that must have had some initial challenges. So how did you even adapt to them?
Sure. So, as I said, the first thing I did was the master's in Massachusetts College of Pharmacy. So I left BCP and went to Boston. I think what prepared me the most was this exposure to so many different subjects. Right. I think that was the most appealing part of this education. BPharm, but from so many different professors. I mean, sometimes you used to get professors like visiting professors from UDCT, etc., or maybe some even industry professors, industry scientists would come and give us a lecture. So that just giving us exposure to so many different topics, probably what prepared me the most for education. The challenge was more about loneliness. Challenge was more about making friends and just being away from family for the very first time in my life. That was a challenge. Not the challenge was not the the studies.
That must be relatable to all the students who are now pursuing in abroad. It is very inspiring that how you navigated through your challenges of initial days. But also heartwarming to know that BCP laid kind of a foundation for this journey.
Absolutely.
So,
yeah.
What are your views about AI and then biologics, which are advancedly progressing? So, how do you see the future of DMPK evolving in the coming decade?
Everybody's trying to see what you can do with AI. Now, I would say that DMPK as a discipline has already been using AI. I mean, when you talk about all these in vitro systems or these in silico models that are built, that's all AI. And we've been using a lot of the in silico models to figure out what molecules to make. That's AI. So, or we've been looking at the structures and deciphering, okay, this is not going to be very metabolically stable. So let's not take this molecule in vivo without doing any studies. That's AI. So that has already been happening. I think more and more with the FDA's approach for a new alternative approach, the NAM that they're advising. Everybody's thinking critically, what you can do in vitro, what you can do in silico, where you can avoid in vivo animal studies to expedite drug development. It's going to take time. It's not going to happen right away. It's going to require a lot of data, first of all, to believe in those models, to validate these approaches before you can use them as an alternative to in vivo animal studies. But certainly, with the advancement of AI, the computer power that is now available, large data sets that, one can manipulate, one can work with, things will get easier, things will get faster. And hopefully that just means that we can bring drugs to patients faster.
Yes, that's the ultimate hope for everyone. So, ma'am, what is your lesson that you have learned throughout your scientific journey or industrial journey?
That's an excellent question, and I wouldn't say I'm necessarily adopting it, but what I would say is my learnings are be curious. As a scientist, you have to be curious, otherwise you're not going to learn. And my second advice would be to don't be afraid of taking risks. I think that's very important. Only when you challenge yourself, only when you think, go outside your comfort zone, you achieve good things. So, uh, don't be afraid of risk taking. Just challenge yourself. That would be my other advice.
That's the great advice. Ma'am, when you think of your BCP days, is there any particular labs or mentor or incident that sparked you the curiosity which you are referring that students should have?
See, it was so long ago, right? I'm not going to be able to answer that question very well. I mean, I remember, I wouldn't say there's one incident or one professor or one lab. Um, I think there are the many. Like I used to like Uday Khanolkar's pharmaceutics lab. Then we had Professor Manik who used to teach us industrial pharmacy. Just snippets here and there. I remember. Professor Bhalla was there, of course. Dr. Tipnis was there. Beyond the professors, beyond the classes, I remember my classmates, the camaraderie we had over the 3 years period, and how we became close to each other. Saturday used to be half day. And then how we would just hang around in the cafeteria and then maybe just suddenly decide to go to a movie. And then many of us would have cars. So we would just pile up in 2 or 3 cars and, head to a movie. And then the whole rest of the day, Saturday would go in that. That comes to me as a very fond memory. And many of them, we are still in touch with those friends.
That's very nostalgic. And ma'am, what were your favorite subjects back then?
I really can say that one subject was my favorite. I think I liked medicinal chemistry. Maybe that is why I ended up doing master's in medicinal chemistry.
Finally, if you had to describe your journey from BCP to AGOs in one word, what would it be and why?
One word I would say exciting. That's how I would describe my journey. You said why? Why is because of all the support I got from my faculty, my friends, my mentors, and now all my teammates.
Ma'am, this conversation has been deeply insightful for everyone. Thank you so much for sharing your experience, your reflections, and your perspective. And definitely, your journey is truly inspiring for our students. And on behalf of Bombay College of Pharmacy and BCP Alumni Association, I would really like to thank you for joining us. And whenever you are in, India, please do visit to BCP. It's your home, and you're always welcome here. And thank you so much for joining for this podcast. We're really grateful.
Thanks, Yash. Thanks for taking the time and thanks for coming up with such intriguing questions. I hope that this will be useful to you and your fellow students.